Mitosis. It explains why a tiny seed eventually grows into a huge plant, why a cut heals, and why a small population of pond protozoa can suddenly explode in size. Today we're going to learn what the purpose of mitosis is, what organisms do it, how it works, and what happens when it goes wrong. Okay, here we go. The overall purpose of mitosis is to create new cells. However, the specifics depend on the context. Mitosis is involved in growth, repair, and sometimes in reproduction. Somatic or body cells undergo mitosis in order to help an organism grow. The cells divide and make identical copies of themselves. For instance, skin cells divide and give rise only to new skin cells, and intestinal epithelial cells in the gut only give rise to new intestinal epithelial cells. Which is good because you probably wouldn't want ciliated columnar epithelial or goblet cells on your face secreting mucus. That'd probably be pretty awkward. Some cells, like mature red blood cells, don't divide at all and never undergo mitosis. Mitosis is also involved in repair of damaged tissues. A shallow cut or a burn will heal over in a few days as new cells divide and cover the area. Some cells, like epidermal, skin, gut, and liver cells, regenerate very quickly. The jury is still out on whether other cells, like heart and nerve cells, divide very, very slowly or just don't divide at all once mature. There are organisms that use mitosis in order to reproduce. These organisms, such as protists, reproduce by cloning themselves. It's important to remember that gametes, sperm and egg cells in humans, are not produced by mitosis. They're produced by a related process called meiosis. We'll cover meiosis in another video. Nearly all organisms do mitosis. This includes all eukaryotes, so animals, plants, fungi, and protists. Prokaryotes, also known as bacteria, do divide, and it looks a lot like mitosis. However, because they don't have a nuclear membrane, technically it isn't actually mitosis. So with that out of the way, let's move on to the actual process of mitosis itself. It's important to remember that mitosis, the process of cell division, is only a tiny portion of a cell's lifespan. Cells are actually in interphase most of the time. The order of phases are interphase, prophase, prometaphase, metaphase, anaphase, and telophase. We'll begin by discussing interphase. During interphase, the genetic material is in the form of chromatin. It will be packaged more efficiently later on. Interphase consists of three parts: G1 phase, S phase, and G2 phase. In G1 or gap 1, the cell grows, creates mRNA, and manufactures proteins. At the end of G1, the cell checks for growth and for damaged DNA. If the DNA has suffered damage, the cell will attempt to repair it or will stop dividing in order to contain the problem. In S phase, the cell grows and it duplicates its DNA. There are now 2 copies of every strand. When the cell divides in half, one DNA copy will be in one daughter cell and the other DNA copy will be in the other daughter cell. In G2, the cell grows and manufactures organelles that are necessary for mitosis. Perhaps most importantly, a set of organelles called centrosomes will duplicate. At the end of G2, the cell checks one final time for growth and for damaged DNA. Now the cell is ready for mitosis. First, it enters prophase. During prophase, the nuclear membrane begins to dissolve so the DNA can eventually be divided. The 2 copies of the DNA are still in their messy chromatin form. However, they need to be packaged efficiently so we can ensure that it divides properly. The DNA begins coiling itself very tightly around proteins called histones to form structures called chromosomes. Chromosomes look like this and consist of 2 chromatids held together by an attachment point known as a centromere. The centrosomes from G2 are also moving towards the poles of the cell. Second, we enter prometaphase. In prometaphase, the chromosomes continue to condense until they are coiled as tightly as possible. The centrosomes continue moving towards the poles of the cell, and the microtubules begin to emerge. Third, we enter metaphase, the longest phase of the cell cycle. The chromosomes are pushed into alignment along the equator of the cell by special proteins called motor proteins, which are attached to the centromere of each chromosome. The microtubules also attach to the centromeres. Later on, these will shorten and pull the chromosomes to each side of the cell. Fourth, we enter anaphase. This is where we will separate the chromosome copies from one another. The motor proteins exert so much pressure on the 2 chromosome copies that they split apart. Each copy is then dragged back towards one of the two poles of the cell as the microtubules shorten. Finally, we enter telophase. The separated DNA copies are pulled to each pole of the cell. The DNA is put to work almost immediately to begin rebuilding and reorganizing the important cell components. The nuclear membrane begins to reappear, and the chromosomes unravel back into chromatin. The cytoplasm also divides in two in a process known as cytokinesis, yielding two identical daughter cells. The cell then re-enters interphase, and the cycle begins all over again. In summary, the genetic material is duplicated during interphase. Then during prophase and prometaphase, it condenses into chromosomes for easy separation. In metaphase, the chromosomes are pushed into alignment on the metaphase plate and then are pulled to opposite poles of the cell during anaphase. The cytoplasm then splits during telophase, and the chromosomes then unravel back into chromatin. We've taken a diploid parent cell and created two identical diploid daughter cells. Phew, so that's mitosis. Now, we have to ask ourselves what happens when mitosis goes wrong. Well, that depends. There are two basic categories of incorrect mitosis: incorrect division of genetic material and too frequent division. Incorrect division of genetic material most frequently occurs during anaphase. Usually what happens is the chromosomes are stuck together very, very tightly, and so they don't separate correctly when the spindle fibers begin to shorten. This results in a condition called aneuploidy, where one daughter cell has too many chromosomes and the other resulting daughter cell has too few. Depending on the type of cell and which chromosome didn't separate correctly, aneuploidy can actually be fatal. Usually, cells divide at a measured pace and stop dividing if there aren't enough nutrients available. However, exposure to certain environmental factors such as drugs, chemicals, ultraviolet light, or just a simple DNA copying error can cause mutations that impact the frequency of cell division. When these cells divide uncontrollably, they can form a growth called a tumor. Some types of mutated cells only form growths in one area, but others will metastasize and begin to spawn growths in other parts of the body. When metastasis combines with uncontrollable cell division, we call it cancer. Alright, that pretty much finishes up our discussion of mitosis. If you found this video useful, I hope you'll consider subscribing to my channel and checking out some of my other videos. Thanks again for watching, and please remember to like, comment, and subscribe.